Thanks very much. Thanks for coming, everybody. I want to thank you all for coming, and I want to thank the organizers of the conference for having us do this. This is my favorite conference, not just at Penn State, but my favorite conference all year, because there's such a wide variety of really interesting topics going on, but what's even better is that everybody's here. This is the one event at Penn State where I can come and see all my colleagues and talk about teaching, not even so much about teaching with technology, just talk about teaching with everybody. And so that's what makes it so exciting. It's really great to be here. Thanks for coming. I'm going to talk today a little bit about a course that we developed in my department, Biochemistry and Molecular Biology, just last year for the first time. It's pretty new, and we learned a lot, and the reason we came was because we would like to share with you what we learned if you were thinking about doing a fairly big inverted classroom, especially if you want to do it in science. The course that we did was pretty idiosyncratic in terms of our needs, but hopefully what we did will apply to what you want to try. And I brought all these students because my view on the course was really kind of attenuated, right? They were the ones on the other side. So we have two students, Abby and Alex at the end, who took part in the class. They were actually students enrolled in the class. And the other four, Jake, Zach, Sarah, and Sophie, were learning assistants in the course. And without them, the course wouldn't have succeeded at all. So to the extent that it succeeded, I relied completely on them. So what I'm going to do very fast is just they can introduce themselves, say what year they were, and their hometown, and just so you can know who they are. So we'll start with Abby.
I'm Abby Coyne. I'm a freshman here at Penn State. I'm a microbiology major, and I'm from Philadelphia.
I'm Alex Arvanitis, also a microbiology major in freshman year, and I come from Kennett Square, Pennsylvania.
I'm Jake Boyer. I'm a senior in biochemistry. I'm from State College. This summer I'm starting my PhD, and so I wanted to gain like some teaching experience, and that's why I became in LA to begin with.
I'm Zach. I'm a senior in biochemistry. I'm from Lancaster, Pennsylvania, and I wanted to be in LA to kind of see the other side of the classroom, to see the teaching aspect of it.
I'm Sarah. I'm a sophomore majoring in biochemistry and molecular biology, and I'm also from State College, Pennsylvania. So one thing at Penn State, in our department, we don't have like a freshman B&B class, so this was a cool opportunity that I wanted to join, being part of a freshman B&B class.
My name is Sophie. I'm a senior majoring microbiology. I'm from South Korea, and I wanted to be in LA because I wanted to learn how I can communicate with others and how to help others.
And they really helped others, that's for sure. Starting with me, the other. Okay, so that's everybody. This is a picture that we took on the last day of class of the course that we developed. It was called Majors Colloquium, B&B 398A. And you can see it was a fairly big class. There were 80 or so students when we started. A few dropped, so there was a little bit fewer than 80. So this was a fairly big course to do a flipped classroom in. Okay, so let me first tell you about the programs in my department. There are three. There's biochemistry and molecular biology, which have options in molecular and cell biology or sort of old school hardcore biochemistry, like Jake did. There's microbiology, and there's a major called biotechnology. And these overlap a lot. And in short, the first 2 years are very similar, if not identical, in all these programs. And the third and fourth year is where they diverge, where students specialize. Okay. So first year students coming into these programs, so this course was for all the students coming into all three of these programs in their first year here at University Park. You find students who come from schools where, frankly, their science and math preparation wasn't so great. And you find students, especially townies like Jake and Sarah, right, who might arrive here having already had fairly significant research experience. So this range of preparation is a thing that the first year courses in science and math here have to deal with all the time. There's about 100 students that come in to our programs, and we got about 80 of them that we could schedule into this course. So let me tell you a little bit about the curriculum, and this is true for all, not just our three programs, but for all 6 of the options in biology and for, let's say, a nutrition science major, or a biological anthropology major, or you know, an animal science major, any kind of bioscience major, not just at Penn State, but at a university anywhere, is going to have this kind of format, where the first 2 years are, and I don't use this pejoratively, the price of admission, right? You're going to take 2 semesters of calculus, you're going to take 2 semesters of physics with lab or more, you're going to take 2 semesters of inorganic chemistry with lab, 2 semesters of organic chemistry with lab, okay? And if you don't take those courses, you're not ready to take the major courses where you really diverge and specialize and really drill down into biochemistry or developmental genetics or nutrition or whatever the major is, okay? And so the bottom line is that there's a lot of delayed gratification. These students have to bust their humps for 2 years before they get to the courses that they really want to do, or so they think, when they arrive at Penn State, okay? And so this was a big need that we were trying to fill. So this is actually, this is what we hand out in advising meetings, okay? And this is, there's no pointer, but this is the BMB major with the MCB option. But again, I want to stress, this would look quite similar for biology, physiology option, or any of the other examples that I've talked about. And again, we'll just zoom in here. So the first 2 years, calculus, chemistry, organic chemistry, physics. You might take a statistics course. We make students take, in the second semester, an introductory microbiology course. But again, in fact, it used to be just 2 or 3 years ago, students didn't get that microbiology course until their second year. So there used to be this terrible donut hole where they would slog through courses in chemistry and calculus without any motivation for why am I killing myself in this class before they even got to their first major course, okay? And then again, in the third and fourth year, this is where you get down to it, okay? And these are where the majors become different, our 3 majors and the other majors too. So what we needed was a course in the first semester that gave an introduction to all these topics that the students were motivated to get to, but also motivate those topics in terms of, you know, at the end of the semester, hey guys, do you think you understand why you might want to take a physics course now, right? Is it a little bit more clear to you that organic chemistry isn't just sadism on our part, you know? We wanted to foster, right? So this cohort that comes in is 100 students, and that's just big enough that I can't really. By the way, I should have said at the beginning, I'm the director of undergraduate studies, so I oversee these programs in my department. And so they're just big enough for me not to be able to meet everybody before they come to the first day of class. And they're just big enough, right? So, you know, the size of this program, you know, we've got 400, 500 students in all the programs put together. That's a rounding error for some programs at Penn State, University Park. But it's just big enough that students don't necessarily get to know each other even 4 years later, right? And so we wanted to do something to kind of foster a group identity. The third thing we wanted to do, and frankly, looking back on it, in hindsight, this was probably too many goals. There was a little bit of a kitchen sink in here. We might have tried to do too much. But we were trying to get students to break away from this high school conception of science as either being biology, or being chemistry, or being physics, but not being anything in between, and not being anything else, and being something where you take the cookbook up to the bench, and you mix the things together, and then you write down what you saw, which is the thing that matches what they told you were going to see at the beginning, you know? Also, we want, right, so there's all this, right, students are really excited about meeting real scientists and doing real research and finding out what that is and getting involved. So we wanted to try to give a little bit of a taste for that in a couple of different ways. Okay. And sort of following along this, when students come, they get this orientation, and the dean tells them, right, like, this is not high school anymore. This is going to be different. You're going to do all of this creative, high-level work. We're going to expect much more from you than you were ever expected to perform in high school in math and science. And it's not going to be cookbook. You're going to be doing, you know, creative work. You're going to have access to research. You're going to be creating knowledge, right? We tell them that, and then you saw the schedule, right? We make them take calculus and organic chemistry, and so we're kind of lying to them, actually, at least in the short term. And, you know, when you're 17, 18 years of age and you arrive here in August, you know, 2 years later, is not something you can even form in your mind. Okay. So, before I was in BMB, I oversaw the programs in immunology and toxicology in agricultural sciences. And I developed a course which was a straight up 3-credit first-year seminar course called VBSci 50. And it was for everybody coming into IID and Tox. There were only about 20 students coming in every year. So that was a cohort where I did know everybody. That was a cohort where they could easily know each other. That was a cohort where they all fit into one first-year seminar. So that was a whole different ball game. What I had them do was pick topics. They gave individual presentations with lots of feedback. I helped them develop these presentations. A lot of one-on-one guidance from me, lots of feedback from peers, and it was boutique-y little seminar. You know, when I say seminar, this is what it, like we all, you know, sat in a little room and talked it over. So how to scale this up, how to make this fit 80 or 100 students. So when we racked our brains, we needed a bigger space, and so discussion was going to be hard if we were in a big auditorium. We had more people, and there were all kinds of things that I didn't even realize wouldn't scale, like my sense of humor, you know? There's stuff that you can get away with in a room full of 20 people that just bombs when it's in an auditorium. And it's just something about the number of people and the space and the distance between, you know, an eye contact and all that stuff. And so that was different. And there weren't enough slots on the calendar for everybody to give an individual presentation, so we had people team up. And then the kind of leap of faith that we took was, since we're going to have these teams anyway, why not just have them, instead of giving a presentation, why don't we have them make something that people can watch outside of class, so that when they come to class, they can have a much more enriched discussion in small groups, so that we can, this was our stab at scaling this up to having a big room with a lot of people, so that people could still have that kind of contact and intimate, well, and also substantive discussion of what we were talking about. Okay, so we used the one-button studio. So how many people are from University Park here? Yeah. How many people are from campuses that have one-button studios? Yeah, the idea is there's a little room, right? And you go in, you plug in your jump drive, you hit the button, it starts recording, you give your presentation, you hit the button again, it saves to your jump drive, and you've got this nice video with, you know, professional lighting and green screen in the background if you want to use it. It's really a fantastic resource. And virtually all of the teams use this. Some of them got creative and did other things, but almost everybody did this. Okay, so here's the syllabus of the course. The goals are not typical, you know, first and second year course goals, right? Get inspired, okay? But a key goal here was to get a molecular perspective, right? When I say diabetes, you want to think of symptoms and not stop. You want to think of symptoms and be able to connect them with beta cells getting wiped out by a virus when you're 9 years old and now you got a bum pancreas, right? You want to connect the symptoms with a lifetime of eating the way I do, and eventually you dig yourself into a hole that you can't get out of anymore, right? And why does that happen? Well, because there's molecules, right? There's hormones, and there's receptors, and there's signal transduction pathways. And don't worry about all of the details of them, but now that you've seen one, okay, because one of your peers, you know, 4 of your peers have talked about it for 20 minutes, now you're ready to fit that into something, hopefully at the end of the semester, okay? So, and here's the second page of the syllabus, and there's our 4 LAs looking cute. You know, when I was there, so when I was 30, 35, when I met students, I just projected my old self onto them, right? And now it's different. When I meet them, I project my kids onto them in 15 more years, right? And so these pictures, I'm a big softie, and these pictures really mean a lot to me. And what you guys did for me really means a lot to me, so thanks. And thanks for being here today. Okay, so anyway, here's the way students earned grades in the course. 30% of the grade was their video, basically. 25% of their grade was what the audience in the room, how they evaluated their in-class summary of that presentation. 25% was the in-class evaluation of the team's leading of the discussion. And by the way, they brought 2 discussion questions. One was about the molecular mechanism, and one was about the impact on human beings or society of that molecular mechanism, whether it was on production agriculture or a drug or a genetic syndrome or whatever it was. Okay. And then 20% of the grade was, and this was a crude, ham-fisted proxy for participation, but there was a clicker question at the beginning on the thing that they were supposed to have watched in the first place. Okay. Here's the schedule. I had a couple of introductory lectures. Then I had guest lectures from, buddies of mine in the department to get them excited about research, which was something that we kind of shoehorned in. We put it at the beginning so that we would stall, basically, so that the teams got assigned randomly, so that the first team didn't have to present, 2 weeks after they arrived at University Park. In hindsight, I think one of the things that will change is not to have so many faculty lectures, at least not front-loaded, and for me to do more than just 2 introductory lectures. The idea of the introductory lectures is just to kick it off and give people an idea of what the basic vocabulary is, get everybody sort of on the same page, but 2 really isn't enough. And next time around, I think I'll sacrifice my buddies being able to come and talk about their labs and give more background so that everybody has a little bit more confidence and a little bit more shared vocabulary. I showed you the grading scale, and I just want to stress that I did none of the evaluations. All of the evaluations were by peers. The most important evaluations, by the way, were within the teams evaluating each other, right? That was the major portion of the grade, and there were some people who quite rightly got nailed on that. And I just thought that it was right and proper, but also it turned out to be politically helpful for me to have that come from their peers at the end, and for me to just be like, look, I'm just the accountant. This is just Microsoft Excel. Okay. The other thing is that I've taught enough discussion courses, actually with small groups, that I know that when we're talking about stem cells or when we're talking about anything that has the hint of controversy, people inhibit themselves from speaking if they think what they say and it's comporting with my opinion affects their performance. So I just took myself out of the equation, and that means that believers can feel like they're part of the group too, in that case. Okay, so here's the rubric. This was for the in-class summary. The whole team got evaluated this way. So what you see is it's brief and polished. It orients the discussion. It's engaging, illuminating, starts and ends on time, and provides sufficient time for discussion. And then each individual who gave the presentation was evaluated this way. By the way, I'll put all these slides on the website. And this is really just kind of nuts and bolts, so I won't take any time going through this. So here's what, let me show you a couple of these. And this is going to be way too loud. This also shows you the rubric that students filled out when they watched the video before class. Sorry, let me zoom in a little. So clarity. Oh, sorry. sorry. Is anything happening up there? No, it's not here. Better? Yeah. Okay. So this is the rubric everybody saw, and you get the video, and then the rubric. You put in who you are, just so that I can make sure that everybody does it. And these evaluations the teams only see the aggregate evaluations. They don't see anybody's particular score for them. Clarity, originality, accuracy and completeness, the video itself, they're sort of intangibles, okay? And this one, I chose this one because it's kind of the funnest. This is going to be way too...
Hello! Today we'll be talking about
Yeah, Kirsten's enthusiasm was what I wanted you to see, but sorry.
telomeres and cancer. And this presentation is given by Mike Brulo, Sally Lee, Ellie Leska.
Okay, so it gets more boring when they start to talk about... Right, so they get right down into the weeds. So they're talking about mechanisms of DNA replication and how DNA replication at the end of the chromosome can't complete, and so you have to have another enzyme called telomerase that fixes that, right? And this is very important for cellular aging and probably for physiological aging. They did a really great job of tying that all together in 15 minutes.
This RNA primer is created.
Okay. So I won't take time to show you the other examples.
So that's a 15-minute video?
I told them that on pain of death, they could not go past 20 minutes, and 15 minutes was the target, and almost all of them did that. I only had to kill one team. Okay. The SRTEs. So Debbie Gill gave a talk in here before us about doing this at Du Bois in Spanish classes. And she showed, she actually showed data of how the SRTs plunged and then sort of declined back up when she took a conventional Spanish course and made it into an inverted class. We changed so many variables here, and this was new, so there's nothing to compare it to. So I regard the numbers as basically meaningless, especially because if you read the open-ended questions, there's passionate hatred and passionate enthusiasm. And so that bimodal distribution kind of got smoothed out in the numbers, and so the numbers aren't very informative, I don't think. But what I found was, I learned nothing in this course. This was stupid. Versus, I loved this course. I'm sure this is the right major. I'm totally on fire for biochemistry now. For the instructor, right? Like, Howell is the best. I love his passion, versus what an asshole, He was so rude, interrupting our presentations to tell us that, we'd had this fact wrong, And so part of this is also to kind of socialize them into the practice of, when we discuss science, you have to have everything right. And if you don't, people aren't going to take your feelings into account first. When we explain that, no, helicase is made of protein, not RNA, So that was a thing. So here's what I think I would do different next time. Oh my God. So for each of these presentations, I met with the team of 4 twice. And getting 5 people in the room at the same time twice was really hard. That was a lot of effort. Okay, I mean, that was effort almost as much as actually helping them in the room. It was worthwhile, but what I think I'll do is make the schedule so that there's a spot in the week so that people set that aside, so that I know I can meet with them every week, okay? We need to have more introductory lectures, there's no question about that. What I think I'll do next year is I will model, or even better, this was one of their ideas. Actually, all the good ideas that were in here, I didn't cite whose idea they were, but any of the ones that you thought was good came from one of the LAs. I had a meeting with the LAs every week where we sort of, did a barometer, like, how we doing, what do you think? And basically every week they were like, yeah, you need to change this thing that's not working, right? And yes?
What did the LAs actually do? I didn't quite get it.
Yeah, so what the LAs did was during the discussions, so the teams would come in, they would give a short summary of what was on the video in order to sort of cognitively orient everybody to the discussion questions that they were going to pose to them. And then I and the teams and the LAs would sort of flip from flower to flower in these groups that had come up to make discussions to sort of prompt them to go through these discussion questions. And what we did in those meetings, besides them helping me fix everything that I got wrong the first time, was to sort of anticipate what the roadblocks would be in answering those discussion questions and try to get ourselves ready to help lead those discussions.
They cast them in these little teams, and they were, what, 5 students or so?
Each team that was presenting had 4 students, sometimes 3, and the discussion groups would have, 4 or 5 or 6 or 8. Okay. So one thing that I think would be better would be to have LAs as a team at the beginning do a presentation so everybody sees what a really good presentation looks like. What I did this year was I don't tell Abby and Alex. So the assignment of teams was random, but it was skewed random. So the students that I knew were going to do well, students that were honor students or that had approached me and made an impression on me for having a lot of initiative and enthusiasm, I put them on the first teams, so that people could see what a good presentation would look like first. The rubrics can be improved. I so regretted having the rubrics be on paper and collected them all, and then I had 80 rubrics that I had to put into Excel. And that was murder. That was a huge mistake. So the rubrics that were online, I just imported from Google Forms, and I really want to do that for the in-class rubrics too. The big thing is just managing expectations, because students come in and they're really ready for a class where they sit there and it's like watching me on TV, and every few weeks they get an exam. That they can handle, right? But this thing where it's like everything was outside of their comfort zone, kind of unapologetically, right? I think there were some students that reacted to that badly. Okay, what we're going to do with the rest of the time is I'm going to defer to them. I want to answer your questions about what we learned, what was good, what was bad, and I think they have a lot more insight than I do. Okay, so fire away. Yes?
So when the teams of students are creating these presentations, are they basing their knowledge, the knowledge that they're going to share in their presentation, are they drawing that from a textbook that they have? Like, where are they drawing that knowledge from that then they're sharing with the class? Does that make sense?
There were 2 meetings with me, followed by a meeting with the LAs. In the first meeting with me, we would kind of talk over, all right, what ideas do you have? Which of those ideas would make a good topic here? And I didn't try to steer them then. And then at the end of that meeting, I wanted there to be a topic picked, and for me to kind of say, well, listen, here's the sort of things that I want you to talk about, because they're going to connect to other topics, and they're points that I want everybody in this course to sort of get before they leave. And then I would print out, I would find textbook pages, or I would find not primary literature, but reviews for them to look through. And then in the second meeting, they would bring slides, and I would say, no, this is a Whopper, right? Like this is made of protein, not RNA, kind of thing. And then in the third meeting with the LAs, they were supposed to bring drafts of the discussion questions that then they got refined by the LAs. So those were the 3 meetings.
So they had a decent amount of autonomy in what topic they chose, or was it kind of from a suggestion?
One thing that we did in one of the first meetings was I was just like, okay, everybody, topics, and I had to screen up with just a spreadsheet, and I made a list of topics that they called out sort of by basic category, and I said, okay, think about which of these you want to do. So it was really blue sky.
Can I ask you guys what it was like to be in class? Like, what were you doing when class was going on?
So we went around and helped them, approach the discussion questions. I think the more informative thing and insightful thing for us was outside of class, like helping them come up with questions and refining the science, really. I think outside of class was, better for a lot of the groups and the LAs, but...
Yeah, we were kind of the quality control. We would go through their entire presentation, because I know every group that I met with, I would catch 3 to 5 facts that I just knew were wrong, and I would tell them, like, oh, this isn't right, we're going to change it. And then their discussion questions, they were kind of very, very broad, and we kind of narrowed them down a lot more. So we really quality control before they actually presented.
So did you get training? And this is great. Pedagogical technique.
We had a course on it. We read some primary literature and had discussions. It was on Monday nights, right?
Yeah.
Monday nights.
So all LAs are required to take a pedagogy course.
Which is that BMB 408? No, what?
That's the
course is
teaching
It-
credit. It's Science 297
Yeah.
C.
Yeah.
All right.
And basically every week we would be required to read like a paper about like teaching techniques and things like that, and then we would have discussion about what do you think is actually happening, like how do you convey and communicate, how do you deal with students who are more stubborn or who are shyer, kind of deal with the range of students also. So that was really helpful.
And that was all through primary literature? Did you have a book?
Yeah,
Yeah.
the professors gave us a
booklet, yeah.
booklet about.
But that wasn't this guy that?
No, I had nothing to do with this.
Okay.
I farmed that out. So the College of Science has a Center for Excellence in Science Education, led by Jackie Borjutinsky in chemistry. And also participating in this pedagogy class for LAs is Meredith DeFelice from my department. And they do a lot of heavy lifting. For all of the LAs in the college every semester, they're required to take this pedagogy course, and it's huge. So that I didn't have to do any of that. And in fact, I benefited from that immensely, because they would come back to the meetings with me later in the week and say, yeah, you know, we read this paper and you're doing this all wrong. You know.
Well, for a topic, I can't quite grasp this discussion. Are they supposed to really know the 15-minute presentation well enough that they can discuss that, what I consider fairly advanced biochemistry, in a group situation? They know the language, they know the concepts, and now you're asking them, give an example of a discussion question that just isn't regurgitating what's on the 15-minute.
That may be a question for you guys.
The 15-minute thing. What did you discuss?
Well, when we were doing our research, that actually was a problem. How much detail we should delve into before it becomes moot for our purpose. And our purpose was to understand our topic well enough to deliver this information to a person who maybe doesn't know enough about this topic. And the amount of material that we were supposed to know in terms of details, like specific proteins and syncable pathways, after a certain point it becomes redundant. So we need to understand the information well enough to ask a well-designed question and understand and be able to explain the answer, but not enough to teach a course on it.
Okay, but it was from that 15 minutes. You didn't have
Yes.
to go read it.
No, there was no extra information.
So
The only
it's sort of like answering an essay question in a biology or biochem class, an essay question to discuss the concept of what is a telomere and what does it do, something like that?
That's exactly right, except the material comes from the 15-minute video and the
15 minutes.
short
So
summary
how many
class.
times did you look at that 15-minute thing to perform well in the class?
Usually just once. It was very relaxed. The course itself was not to put pressure on the students, but to excite them about biochemistry, get them interested, facilitate conversations between fellow students, and not scare them away from this major.
Oh, good.
And also with the discussion questions, they turn into more application-based. So instead of just instead of just repeating what we just learned, it was how can you take this and apply it to new research techniques, and how can this problem be solved? Like brainstorm this and try to come up with new innovative ideas to develop new scientific techniques for things.
And that's where the LAs came in. They would come around and monitor if we had good intentions and the idea was creative, but if the mechanisms were completely
They
wrong.
would listen in and coach
Mhm.
you a little bit during the 15 minutes.
That was always fun, yeah.
I would say I was incredibly impressed with what they came up with, definitely.
Wow.
Did you have any kind of learning objects that you brought with you to class? Like, did you have a pathway or some kind of image? Or was it just you watched the video and came in and then did the discussion sheet?
As an audience, as a person
Yeah,
in
as
the
an
audience,
audience.
yeah, there was no. We would just watch the video. Sooner or later, after there was a problem in which many of the students were not watching the videos, Dr. Howell started implementing quiz questions that would be graded for points. So it would stronghand or browbeat people into actually watching the course. It was a good idea.
Well, and the other thing is that there were some groups, when they had a fairly complicated pathway, that would give me a PDF that I made 80 copies of and brought to help
Seems like
people
it would
consult.
Help, yeah.
And so you would watch the video outside, but in the beginning of class there would be like a summary of the presentation just to make sure that, to kind of bring everyone to base so that they have some time to foundation, so that when we do go to the discussion question, they have some knowledge too.
How was the summary presented? Just in PowerPoint?
Yeah.
Yeah,
It's a
PowerPoint.
synthesized PowerPoint and presented under Dr. Howell's guidance, and he would point out if anything was incorrect or needed clarification or if something was vague.
So did students take notes? Like, did you guys take notes while you were watching them?
In the beginning, I actually took notes, and then I realized that taking notes is not necessarily the best thing to do.
And
Sorry, go ahead.
And also at the beginning, so you would have, the groups would have to give like quiz questions too that were less mind-boggling, more like, do you know what I presented? So
Right.
that was also good to create some knowledge foundation. And then at the very end, we would have the discussion
So
questions.
there was a little quiz after this presentation.
Yeah, at the very beginning.
Was with clickers or something?
Yeah, exactly.
Yeah, clicker quiz, as soon as we sat down. Right. In fact, the clicker quiz happened before the summary. So you'd walk in, and as a way just to make sure that this was imperfect, but they'd watch the video, yeah.
Yeah.
So just a question of fact that you wouldn't be able to know if you hadn't watched the video.
Wow. You are smart guys. I am impressed.
You had a question, and then back to you.
Are we talking a 15-minute classroom, 75-minute classroom?
50 minutes, yeah.
Okay, so can you just tell us what each portion, how much time each portion?
I'm trying to, this should have been a slide. You guys probably remember better than I do.
The first question, the clicker question, was it was no more than 5 minutes, and the summary was about 15 to 20 minutes. We try to get it to 15, so we would have 15 minutes available for each discussion question at the end. So it ended up being like the perfect 50 minutes.
And how many discussion questions?
Two.
One or two, depending on time.
One or two,
Yeah.
okay. And then, did something get passed in with this discussion? Did they write something on a piece of paper?
No.
Oh.
At the end, we would all kind of come together and be like, so what did you guys think? And specific groups would raise their hands, say, like, well, we were thinking this, and then we would bounce back ideas. So there was kind of a conclusion at the end of each discussion question.
So did you think there was these two guys, slackers in your groups that did nothing?
Well, not necessarily. Well, in my group, I was lucky enough to get two other very hardworking students. But based on the presentations and the questions they asked and their attitude during class, you could tell that there were some really, really disinterested students that did not care, and that made me angry.
This little discussion, that 15-minute discussion.
Yeah, because the discussion isn't. After a while, it became, meet with this group and discuss amongst yourselves. But before that, it was discuss with the people around you, or talk to the person next to you. And if you were talking, or if you're sitting next to someone who's sleeping.
So there's no accountability of that 15-minute discussion, none?
That's right.
Is there a peer review? of
Well,
that?
there was no peer rating of the other people in your discussion group. And to be honest, this is sort of a holdover from, when you have a good old-fashioned lecture course, a lot of us, at least in science, have this sort of mentality, well, you can lead a horse to water, And I showed up, and, you can listen or not, and, we'll see how you do on the exam. But Alex made this point better than I did. Really what we were looking for here was not another course that would give you exam anxiety and have a final. We wanted to give you a course that was meant to really motivate people.
Right.
And it was more about the anthropology of our programs than it was about, particular learning objectives in the program, right? And so we wanted it to be kind of low stakes.
Yeah.
And actually one kind of gap that you've identified here is that, there were some people who, and you couldn't do this in a room full of 20 people, right? But in a room full of 80 people, there were people that slept, And, it's the
University Park, that's the way it is. You can't capture it.
well, but this mentality that I myself was socialized into, you can lead a horse to water, that's something that I have to overcome. And if I want to do a class like this, I have to be able to think of ways to keep people awake.
Yeah.
So we as LAs tried to seek out those groups of kids that didn't seem to care. I had a great time talking to the star students in the class, but I tried to sort of move to the disinterested students and try and reach them in some way. Didn't always work. But
Was there at
sometimes
least
it did.
attendance?
Well, there were, remember there were the clicker points at the beginning.
Oh, the clicker point.
Yeah.
Okay. Got it.
And so that was basically attendance and also a crude and probably not so effective way to make sure people watch the videos.
Yeah. But they could sneak out after the clicker?
No, you could, well, because, right, so the way it worked, so this was a very dynamic classroom where I would come down and say, okay, everybody, okay, everybody, all right, here's Alex and Abby and two other people, and they're going to talk about cystic fibrosis. All right, let's welcome them. And I would go sit in the back. And so I could tell who was on Facebook, right? And I told them, look, like there's no Facebook column in the spreadsheet. So you can do Facebook. But I'll change my opinion about you if I see you doing Facebook every day, if that matters, because I want there to be rapport between me and you. I want you to think of me as a human being, not as this robot that, just spews out lectures. Yeah. And I would attempt when I had a question or when a presentation was starting to drift, I would raise my hand and wait to be called on. I was just a member of the audience, right? Now I was the dominant member of the audience, right? But I tried to give the floor belonged to the team at every meeting. And somebody asked for examples of this kind of question and that kind of question. So, for telomeres, right? What would happen if you were missing this enzyme, right? Or you were missing this component, so that they really understood the mechanism. And the human question would be, would it be great if we could make it so that in all cells, in all tissues, telomerase was always active, right? And of course, students would say, sure, that we'd all be immortal. Well, okay, then what? Well, it turns out that we probably wouldn't all be immortal because the clever ones had caught this kind of aside in the presentation that telomeres are, highly active in cancer cells. So there's probably, right, there's pros and cons to this, just like there is in any biological mechanism. So just to again, that's a good example of directly connecting molecules and human impact. But I think we only answered one of your questions.
Yeah, in addition to watching videos outside of class, did they have to do any sort of readings to prepare for those discussions, or was all the information just coming from?
Completely hermetically contained within that video. And again, the idea was, we wanted this to be low stakes. Like you were going to bust your hump on your presentation, and for the rest of the time, we only asked that you show up and participate. Because, we added this to that first semester, which is already enough. So that's how we designed that.
Is it 3 credits?
It was 2 credits. It was meant to be right, so in the College of Science, there's a 1 credit first year seminar, which is just kind of academic advising orientation for 15 weeks, which is valuable, but we wanted to add content to that, and so we just rounded up to 3. Did you have a question back there? No? Yes, you had one.
Well, this kind of goes along with what you were saying, so the information they were kind of responsible for the discussion was within the video. Was there any formal mechanism for them to ask questions before coming to class if they were not clear on some of the concepts, or did that not seem necessary, or? If the material wasn't clear to them before they got to class.
Well, I'm sure that most of the material was not clear for most of the students after the videos. And so the aim of this synopsis at the beginning was to clear up those questions.
Well, I guess I'm thinking more like, I could see someone very like grade conscious, might be very anxious about these clicker questions at the beginning of class and think like, oh, I watched this video, but I'm not sure if I understand it. Maybe I'm going to do poorly on the clicker questions. I guess I'm thinking of that highly motivated student that like really wants to get every question right.
The PreMed.
Was there a need for kind of a mechanism for them to address any lack of clarity in the content, or was that didn't seem really necessary?
Well, for me, this class was sort of a break from the inorganic chemistry and the calculus and the boring history class. So I enjoyed going through the biochemistry of this. So if I didn't understand something, I just rewind the video in the comfort of my own small dorm room and rewatch the video and see if I could really understand it. But you have to consider the source. If the students who created the presentation didn't understand the material and just kind of wanted to rush the presentation along, then I'm up a creek. So.
And usually the clicker questions were very fact-based. So if you watched the video and you just listen to it, you'll recognize the information. It's not really anything that you really had to truly understand as opposed to just know.
So the other things you might have to understand might be addressed in the discussion. And
Exactly.
Then
Yeah.
that makes sense.
And this is for Alex and Abby. Did you guys tend to sit with your group that you worked with in
Yeah.
class?
Towards the beginning, yes, I always sat right next to my two partners. And then after I started talking to other people, I made more friends. I would sit with them all the time. And then it became a really fun, it became, like I said, a break from all the other courses. And we got together and talked about biochemistry. It was fun for me.
Yeah, I always sat with my group. My group was of 3 people, and we had one person kind of disappear after our presentation. He kind of floated in and out, and we never really saw him. But me and my other group member, we kind of got really close, and we're really good friends, and we always stuck together, and we always sat and discussed.
Okay. Yeah, I was just curious whether stable-based groups, like if you worked in your discussion groups throughout, but it sounds like there's some advantage to meeting other people in the class as well.
Oh, yeah.
Do we have time for one more question? Yeah, one more question.
Yeah. Were the discussion groups then forced? I mean, you said, would anyone sit alone because they didn't know how to join a discussion group? I find that in my class. So beforehand, did you know you had someone to talk to?
They did, but we tried to distribute it, so why don't you go over
Pardon me?
there or something like that. We would point them in the right direction, so to speak. Like if somebody's sitting alone, we'd say, go over and join that group or something like that.
Well, that was sort of your role to
So
get.
you guys
In a
helped
way,
with
yeah.
the discussion as well.
So
Yeah.
there's no one ever sitting just over there?
No, we wouldn't let that.
In the beginning, it was just talk to the people around you, and then we would try to force groups. And then towards the second half of the class, we would combine the group that you worked with another group. So then it kind of forces you to actually get together with people and come together.
This all happened in a room like this, so there was lots of talk and noise.
It was a big auditorium.
Yeah.
Yeah. Yeah, so an auditorium that would fit 120. I purposely got a big one so that people could spread out and get a little bit more comfortable.
Hard to get those rooms when you don't have enrollment.
Yeah,
I'll talk to you, how do you get that?
I cheated.
That's a great idea, thank you very much.
Well, thank you.
You guys are remarkable.
So I want to thank all 6 of you, especially the LAs, for all the help that you've given me in this course. I want to thank you all for coming, and I really hope that there was something today that you can take back to your classrooms and use. Thanks very much.
Thank you.